The aim of this procedure is to assess axon guidance defects in the chicken neural tube after downregulating gene expression in a cell-type specific, traceable manner. This is accomplished by preparing fertilized chicken eggs for in vivo electroporation by cutting a window in the eggshell. The second step is to inject and electroporate DNA constructs encoding microRNAs into the neural tube. Next, the electroporated spinal cords are dissected and fixed in flat mount open book preparations. The final step is to inject a fluorescent dye to visualize the axonal projections. Ultimately, fluorescence microscopy is used to show both the cells experiencing gene silencing and to assess the subsequent effects on commissural axon guidance.
The main advantage of this technique over existing methods is the cell-type specific silencing of genes. To make the plasmids, check the written protocol for instructions.
Incubate the eggs at 38.5 degrees Celsius and around 45% humidity until the desired stage of development is reached. To study axon guidance of commissural neurons, incubate the embryos for approximately 3 days until they reach Hamburger and Hamilton stage 17 to 18. Once the desired stage of development is reached, and before the protein of interest has accumulated within the embryo, remove the eggs from the incubator and place them in a stable horizontal position for 20 minutes to reposition the embryo on top of the yolk at the upper side of the egg. After the embryos have repositioned on top of the yolk,